Nancy. So again, my name is John Kelly. I'm from Loyola University in Chicago. I want to mention my collaborators in this project. It includes Emma Rosie Marshall, who works at the Carey Institute for Ecosystem Studies in New York, and Teresa Chow, formerly of the ISTC, and now Monty Wilcoxon here at ISTC. So my project, we are right in the middle of a 2-year grant from ISTC, so this is kind of my midterm project review. So I'm only hopefully getting my midterm grade today, not my final grade today. For all of those of you who are teachers like me, those are important things for students to recognize. So by pharmaceuticals and personal care products, everyone knows what this means. Prescriptions and non-prescription drugs, including antibiotics and lots of other compounds like analgesics, antihistamines, antacids, hormones, caffeine, as well as a lot of household products that sometimes include antibacterials and antimicrobials. So we know that these things are being released to the domestic wastewater stream. So when we use a lot of these things, whether they are excreted through our body when we're taking antibiotics or analgesics or caffeine, or if they're being released directly and for use as soaps, these things end up in the domestic wastewater stream. And we know there's data that's detected these things in domestic wastewater. Now, when this wastewater enters wastewater treatment plants, a lot of these things are fairly well removed by wastewater treatment plants. However, even when you say they're well removed, the removal is generally not 100%. So it means there is some low-level release to streams and surface waters. And our concern here is that if some of these compounds are recalcitrant, if they're resistant to degradation, even if there's a low-level release over time, it could result in significant accumulations in the ecosystem. The other issue is that some of these compounds, while they're removed by wastewater treatment systems, they're not necessarily degraded. Many of them partition specifically to the biosolids or the sewage sludge because they're not terribly water soluble. So in this case, the sewage sludge or biosolids can be another route of entry of these compounds into the ecosystem. Lots of these biosolids are land applied, especially here in Illinois. When these are land applied, there's the opportunity then for those pharmaceuticals to enter the soil or potentially through leaching to again enter surface waters. So there's the potential for these compounds to enter the environment, although probably in relatively low concentrations. However, they have been detected in streams and rivers in the United States. So there's a well-known study by a group at the U.S. Geological Survey that was published in 2002, where they surveyed for a suite of 95 different contaminants, including a number of pharmaceuticals and personal care products. They surveyed 139 streams in 30 states, although they intentionally biased their survey towards streams that they thought would have the potential for contamination. So streams downstream of wastewater treatment plants or downstream of agricultural areas. Then again, they also analyzed only the water. No sediment samples were analyzed. They found 82 of these 95 contaminants they were looking for, and they found some contaminants in 80% of the streams. So 80% of the streams had at least some of the contaminants from their list. However, the concentrations were generally very low. So 95% of the concentrations measured were below 1 microgram per liter. So low concentrations, but detectable concentrations of these compounds. So the ecological impacts or ramifications for the release of these compounds is really an open question. There's only beginning to be interest in this area. We know that these PPCPs are biologically active. That's the entire point of their use, right? They interact with cells in some way. That's why they're useful to us as pharmaceuticals. So it's reasonable to hypothesize that they may have some biological effects when they're released to the environment. But what these effects are, again, we really don't have a ton of data. We need to understand what categories of organisms are impacted by these things, going from microorganisms to macroorganisms. We need to understand the differences between acute and chronic effects. We also need to understand potential effects on community composition and ecosystem processes. So these are the kinds of topics that we're trying to address in our project. We picked one particular compound, triclosan. So this is a potent antibacterial compound. It was first discovered in the 1960s. At that time, it was used primarily as an antiseptic or a disinfectant in hospitals. And its mode of action, we know now, is that it blocks lipid synthesis in bacteria by inhibiting the activity of this enzyme, enoyl acyl carrier protein reductase. So bacterial cells can form lipids, they can form cell membranes, can be toxic to bacterial cells. So again, when it was first discovered, primarily used in hospitals. But in the 1990s, there was an explosion in the use of this compound in the consumer market. So between 1992 and 1999, 700 products containing triclosan entered the consumer market. So you can currently find triclosan in lots of consumer products like soaps, detergents, cleaning products, toothpaste, mouthwash, even paint. So all of these things, again, are meant to give these products antibacterial properties to inhibit bacterial growth. And, you know, if you go to the supermarket now, at least where I live in the supermarket, it's hard to find any soaps that are not antibacterial now. It's become so popular, these hand soaps and dishwashing soaps, almost all of them contain triclosan or triclocarban, a closely related compound. They've also developed mechanisms to embed triclosan in both plastics and textiles. So you can buy products ranging from plastic cutting boards to child's height chairs to other plastic compounds. Supermarket shopping carts often have triclosan embedded in the plastic handles to prevent the transmission of bacteria, reportedly. They're also embedded in textiles, so you can buy socks and underwear with triclosan embedded in it as well. So there's lots of it being used. In that Kolpin survey from 2002, triclosan was one of the top compounds they found most frequently. These are the most frequently detected compounds they got in this study include this insect repellent, caffeine, not surprisingly, triclosan was found in 58% of their streams, and a fire retardant. Now the concentrations of triclosan in the water were relatively low. The highest value they found was 2.3 micrograms per liter. The median value was 0.14 milligrams per liter. So low concentrations in stream water. So the question we're trying to get at, and the question you may all be asking yourselves now, is why do we care about triclosan in streams? Why should we be concerned? Well, there's a number of reasons why we think we should at least be concerned enough to look and to ask questions. One is that triclosan has low solubility in water and it's lipophilic. So our concern here is that when it's released into streams and rivers, it's going to partition to the sediments. It's going to quickly leave the water column and end up in the sediments. And in fact, a number of studies have found triclosan in the sediments of lakes and estuaries. It's also been shown to persist in the environment for significant periods, especially under anaerobic conditions. So the combination of those two things, it's going to partition to the sediments. Sediments are generally good anaerobic environments, create the opportunity for triclosan to persist. So our concern here is that even though the levels being released into the water are very low, if it partitions to the sediments and can persist in the sediment, there's a potential for significant concentrations to build up in the sediment. And then, of course, we're concerned if you're a microbiologist like me, right? Triclosan is an antibacterial compound. So we're releasing a potent, broad-spectrum antibacterial compound into stream and river sediments. Within these stream and river sediments, bacteria are important components of the ecosystem. They're important contributors to ecosystem function. They play an important role in organic matter decomposition and nutrient cycling, recycling of nitrogen in particular. They produce biomass that's part of the food web of these ecosystems. They also participate in denitrification, as was mentioned in the earlier talk, which can help to ameliorate nitrogen pollution, which is another big problem in Illinois. So bacteria are important components of stream benthic ecosystems. We potentially are applying measurable amounts of an antibacterial compound to these ecosystems. What are the impacts going to be? We know that bacteria can develop resistance to triclosan. There were a couple studies done in Stuart Levy's lab at University of Massachusetts where they showed that if they grew E. coli in the lab with very low levels of triclosan, that they could raise and develop triclosan-resistant mutants of E. coli. So long-term exposure to low levels of triclosan resulted in triclosan-resistant mutants, and they tracked down the specific mutation that gave them resistance to triclosan, and it was a mutation in that gene that's responsible for the enzyme that's used to produce lipids, that enoyl carrier protein reductase. So a mutation in that gene led to triclosan resistance. And again, the key point to keep in mind here is long-term exposure to low levels of triclosan, which is what we think we might see in the environment. Yes?
Low level meaning?
Oh boy, I didn't, I can send you the paper. I didn't write down the concentration that, Stuart Levy's lab used. I don't know off the top of my head. There's also been a number of studies that have drawn some links, although this is kind of open to debate, and there's some arguments on both sides of this issue that exposure to triclosan may also result in increased resistance to antibiotics, so to medically useful antibiotics. A number of resistance mechanisms can be applied both to triclosan or to antibiotics, so there's some concern that if we increase exposure to triclosan, we may be driving resistance to other to clinically useful antibiotics. And this has been shown in 2 different bacterial strains, Pseudomonas aeruginosa and Escherichia coli. So in the laboratory, they could show that exposure to triclosan in these bacteria led to increased resistance to antibiotics. So another good reason to be a little bit nervous about this. So the questions that we wanted to address in our project were, are there detectable levels of triclosan in water and sediments in Illinois rivers and streams? And we were especially interested in sampling the sediments to see if our concept of the triclosan partitioning to the sediments is realistic. Are the levels of triclosan in Illinois rivers and streams impacting the resident bacterial communities? And how does triclosan impact the composition and function of these communities? So we took a 2-phase approach to trying to answer these questions. We're doing field surveys at rivers and streams around Illinois, and also running some artificial stream experiments in the artificial stream lab at Loyola. So we have these 10-meter-long recirculating racetrack-style artificial streams in our department. We have 48 of these streams, and so we can run well-controlled replicated experiments that we dose with a particular compound, in this case, triclosan. So I'll start out with the artificial streams. So in the last year, we've run a number of pilot studies with these streams. Not too many of my colleagues at Loyola have actually used these streams to study sediment processes. They've used them a lot more for looking at biofilm growth on tiles, and so they don't typically try to study things in the sediment. So we needed to try to figure out how to design a realistic-looking sediment for our artificial stream. We did a number of pilot studies to get to this point, and I won't bore you with the details of those. But the system that we came up with seemed to be working pretty well. We mixed sand and pea gravel in a 4-to-1 ratio. I had 200 grams of shredded leaf material, and then 100 grams of sediment from the Nipperson Creek in McHenry County, Illinois, which we took to be a relatively low-impacted, quote-unquote, closely pristine stream that didn't have a lot of human impact. And we monitored weekly in these artificial streams the bacterial community size via heterotrophic plate counts and the triclosan resistance, which we just measured by growing the organisms on agar plates amended with triclosan. And we then, after a while, added triclosan to these streams. So here's our data for the bacterial counts. So the x-axis shows the weeks, the y-axis shows the bacterial cells per grams of dry sediment. So you can see the 2 streams, stream 1 and 2, there was a little bit of movement around. One thing we discovered is there's a ton of heterogeneity within these streams, depending on where you sample. So you've got to be careful and have a good sampling regime to make sure you're not being too influenced by where you grab your sample within the stream. But there's not really much of a difference in our 2 replicate streams, and the numbers were hovering around 2 to 3 times 10 to the 5th cells, which is, close to what you'd see in a natural stream. Around week 8, we added triclosan. We added a high concentration of triclosan, 16 nanograms per liter. This was about 4 times higher than the highest level that they saw in the Colton paper. We picked a high concentration for this pilot study because we just wanted to see if we could get an impact, if something would happen. And again, the Colton paper was only measuring water concentrations, not the sediment. And we're interested in how this triclosan is going to partition to the sediment. So we tried this as our first go-round. So in week 8, we added triclosan. And you can see, after week 8, so stream 1, the one that's shown there in red, that's when we got the triclosan, there was no real decrease in bacterial numbers. So the addition of even that relatively high level of triclosan didn't knock down the bacterial communities really in any noticeable way. However, when we look at triclosan resistance, so here's week 8, 9, 10, 11, 12. This is where the triclosan was added. This is the percent of the community that's resistant to triclosan, that can grow in the presence of a high triclosan concentration on the agar plates. And you can see what's happening in the stream that received the triclosan. We're seeing this dramatic but gradual increase in the percentage of the community that's resistant to triclosan. So our treatment while not impacting the total size of the community, is driving an increase in triclosan resistance. Now, this, of course, raises lots of interesting questions for a microbiologist like me, right? What's actually happening here? Is this a population shift, right? Are some bacterial species dying while resistant ones increasing in size to keep the total population the same? Or is there some transfer of resistance genes from resistant populations to other populations? And these are questions that we can answer later with molecular approaches. So our next steps with the artificial streams, yes?
What about stream 2? What's going on there?
So that one did not receive any triclosan. So its resistance level is hovering around 0.5 to 1%. I don't think those changes that you're seeing, that little bit of a dip, is anything significant. I think it's just going to kind of gradually move around. And again, it has to do with the sampling and the heterogeneity of the system. Yes?
The dosage level was 16 micrograms or nanograms per liter? Because I think the slide that you had earlier, maybe I thought I saw 16 micrograms.
Yeah, 16 micrograms per liter.
That's like 16,000 nanograms per liter.
Right, so this is what they found in the Colpin study, so 2.3 micrograms per liter. We were about 8 times that. Is that what I said, or did I say it wrong last time? Yeah, I mean, you could put it in nanograms per milliliter or micrograms per
I think it was nanograms, you said 16 nanograms, that's what I heard.
Nanograms, micrograms, what's the difference? I'm sorry. So we were within the range of what Colvin saw, but 8 times higher, or significantly higher. But again, it was just a pilot study, it was not replicated. The idea was just to see if we could see any kind of a response. And again, to get our system working well, we knew how much sediment to add, how much organic material, were the system relatively stable over time. So we're happy that we've answered those questions in a reasonable way, so that now we're prepared to go on with a more well-designed study. So this is going to start about a month from now. We're going to have 5 streams per treatment, 3 treatments, no triclosan, low triclosan, high triclosan. Now, I'm sure your question for me is going to be, what's going to be the low level, what's going to be the high level? Well, we want to set those based on what we see from our field survey. So I'll come back to that in a second. So what we want to do is pick environmentally relevant levels that we see in sediments based on our field survey. So those values are still open to our decision. And we're going to monitor bacterial community size and resistance. Again, we're also going to measure activity in terms of both respiration and denitrification, and then community composition using molecular approaches to see if we see shifts in bacterial community populations. Now, our field survey, some of it's been done already and some of it is still ongoing. So in terms of the sites we've sampled to date, we have one non-impacted site that we've sampled so far, Nickerson Creek, which again is located in McHenry County, Illinois. That was our non-impacted site. Our heavily impacted site is the North Shore Channel in Chicago that receives effluent from an urban wastewater treatment plant. Our moderately impacted was a suburban site in DuPage County, Illinois, so the west branch of the DuPage River that receives inputs from a suburban wastewater treatment plant. And we've done so far bacterial community size and resistance. We're still working on respiration, denitrification, and community composition and triclosan concentration. So that data I don't have to show you yet, but I can show you our accounts and the triclosan resistance. So our non-impacted site, again, Nickerson Creek up there in McHenry County, Illinois, right here's Chicago, here's McHenry County, close to Wisconsin. Here's a nice pretty photo of our clean stream. And our total cell counts were 3.7 times 10 to the 5th, which again is on par with what we saw in our artificial streams, about 2, 3, 10 times 10 to the 5th cells per gram. And our triclosan resistance, try to remember this one, is less than 1%, so 0.66% of the cells could grow on triclosan-amended plates. Our highly impacted site was the North Shore Channel. So for those of you who aren't familiar, here's the Chicago River, the city of Chicago. Here's the North Branch of the Chicago River. Here is the North Shore Channel. So the North Shore Channel connects Lake Michigan up here near Wilmette to the North Branch of the Chicago River, which eventually feeds into the Sanitarium Ship Canal, Des Plaines River, Illinois River, Mississippi River, and Gulf of Mexico. So this red star represents the north side wastewater treatment plant. This serves over 1.3 million people, including the north side of Chicago and the northern Cook County suburbs. It has a design capacity of 333 million gallons per day. So it's a large urban wastewater treatment plant. So what we saw here when we sampled the North Shore Channel, both upstream and downstream of the treatment plant, huge difference in the numbers of heterotrophic bacterial cells we could count on our plates. So a dramatic decrease in cell numbers downstream of the plant. I was really surprised about this because there's a lot of nutrients released by wastewater treatment systems.
Is that chlorinated?
Excuse me?
Is it from the wastewater treatment plant when it's released? Is it chlorinated? I mean, they have to chlorinate
I believe
in
it.
summer months.
I don't think
so.
I don't think it is, but I
It
think
doesn't disinfect at all.
I'm sorry, what was it?
The Metropolitan Water Reclamation District doesn't disconnect, doesn't use chlorine.
They don't use chlorine. So they just put that without disinfection, they can put it on?
Yeah, it's a big fight now whether they should or not, and they're resisting it.
Yeah, so again, I was surprised at this. And then to triclosan resistance, upstream versus downstream, although these numbers are close, this is statistically significant. So there was a significant increase in triclosan resistance from the upstream side to the downstream. And you can see our resistance levels here were around 2.5% to 3%, whereas in Nipersink, we were less than 1%. So significantly higher levels of resistance here at the North Shore Channel. One thing that may be playing into this difference not being that strong is the fact that the North Shore Channel doesn't have a whole lot of flow to it. So the headwaters of this stream is a lock here in Wilmette. So there's not a lot of flow. So when the stuff comes out of the treatment plant, there is some movement of it in both directions, both upstream and downstream. So that may be contributing to the lack of a strong difference here, although the numbers are dramatically higher than what we saw in Nipersink. And our other impacted site, our model impacted site, was in DuPage County. This receives effluent from the West Chicago wastewater treatment plant. This has a design capacity of 20 million gallons per day, so it's more than 10 times smaller than the north side treatment plant. So a much smaller treatment plant in more of a suburban setting. This is the treatment plant right here. This is the west branch of the DuPage River right here. So again, we saw a significant decrease in our bacterial counts upstream versus downstream. I don't know if they chlorinate. I should definitely find that out. I don't think that they do, but I will check on that because that would obviously be an interesting thing to know. So again, a significant decrease in bacterial counts. We saw again a trend towards higher resistance upstream versus downstream, but because of the variability here, this one was not significant. So we didn't see a significant change in resistance, although again, at both sites, it's significantly higher than what we saw at Nipersink. So the field survey, we've finished those 3 sites so far. We've done community size and triclosan resistance. We're still working on activity, composition, and of course the triclosan concentration, which will be a key feature to measure. We're also planning to sample a few other sites. We're going to do another moderately impacted site in DuPage County. This is the east branch of the DuPage River, which also receives input from a suburban wastewater treatment plant. And we're also hoping to do a couple of agricultural sites, although we still haven't totally picked the ones that we're going to use. And our goal was to find ones that receive biosolids amendments because of that potential route of exposure from biosolids as a transport mechanism for the triclosan. So that's all I got for today. I want to again thank my collaborators, Emma Rosie Marshall, Teresa Chow, and Monty McClaxton. 2 of the students worked on this project. Brad Verby's a master's student in my lab, and Deanna Diavilla is an undergraduate student. And of course, I want to thank ISTC for the funding for this project. It was very exciting for me to get this grant, and I think it's a really, hopefully you do too, hopefully I think it's an interesting project. Thanks very much.
How about the effect on algae? Some people have said that triclosan is photodegradable.
Right, so there is some idea that it is photodegradable, and that there is data indicating that again. And there is some data that does have significant effects on algae. There's a paper out recently, I can't remember the author's name, but I can send it to you if you want, where they saw shifts in algal community composition in the lab when exposed to triclosan and a decrease in algal species diversity. So it does seem to be having some effect on algae as well.